All right, Justin, I got it. It is on YouTube, so anything you say or do will be recorded. Just so
Wonderful. Sure.
Good luck.
Thanks.
Oh, that looks beautiful.
Thank you very much. Good to see you, Tally.
Yeah, same here, Justin. Neat to see you coming in here.
Oh, thank
Looks
you
like
very
you
much.
already got quite an audience.
Yeah, it looks like we're getting good turnout, so that's great.
Cool. I'm going to mute myself, and things are going all right where you guys are?
Yeah, I'm now in Toronto, so it's pretty quiet over here.
Ooh. How long have you been there?
I moved here in August.
Wow, congrats.
Oh, thank you.
So
yeah, very, very quiet here, but we are on lockdown, so it is
Oh.
pretty restricted in terms of what we can do, but our university is still open, so still able to go to the lab.
Cool. Well, I look forward to your talk.
Thank you very much. Good to see you.
Mm-hmm.
Okay, we're going to start maybe 2 minutes late, just to let everybody come in. I'm admitting them right now, just so everybody knows.
Hi, Justin.
Hey, Laura, great to see you. Thanks for attending.
Yeah, we're looking forward to it.
How have you been?
Good. Pretty, I mean, as good as one can be in the current time. Getting excited.
Are you stateside yet or no?
No, in April, I'll be moving.
Oh, very cool.
Yeah.
Yeah, you're going to be joining a really great group, so it'll be really exciting to be there.
Justin, you're actually attracting Chuck Reback, who I haven't seen in these.
Yeah, I saw that. I got a little bit nervous when I saw that name.
Okay, so I think people are going to keep coming in, but I'll try to let them in late as we go. But we're going to get started. So hello, everybody. Happy New Year, and welcome back to the first NextGen Epilepsy seminar of the 2021 new year. It's no longer 2020. Thank the Lord. This series, so just to give an idea of this series, so this was born actually out of my random channel on my Slack, my lab Slack account, and something that we wanted to do to give back to the trainees in epilepsy research. So something to promote them, you know, I took it to Tally and we discussed it as a sort of an epicenter, UCI epicenter sort of sponsored event, and it's been very successful so far. I think it's going to continue to be successful. So we're very excited for the rest of the year. And so today we have Justin Botterill, and he is a postdoc, actually a postdoc at University of Toronto. He just moved, but he's going to talk to us about some of his postdoc work with Helen Scharfman, working on Mossy cells and dentate gyrus function. And hopefully he can finally tell us what Mossy cells do. I don't know, I guess TBD, we'll find out at the end. And so I'm very excited about this. Justin was sort of born and raised in Canada, did his bachelor's degree and his PhD at University of Saskatchewan. I was complaining about the cold weather here this morning in Southern California, and I no longer feel bad about that. So, and then some of this work, I think, Justin, you've pre-printed it, is that right?
Yeah, we have some new insights on that.
And then there's one of these papers is going to be published or is coming out soon.
It just got accepted on Monday
Cool.
for publication, and the other one was just resubmitted, and we're about 2 weeks since we've done that, and it was under minor revisions, so hopefully soon.
Cool. So hopefully you guys get to see this in advance. So great. So Justin, we're excited to have you. Take it away.
Sure. Thank you very much, Bobby, for the kind introduction, as well as to Momoko and for Tally for organizing this. It's just been a fantastic series so far, and it's a real honor to be part of it. As Bobby mentioned,
all Bobby.
as I mentioned, I'll be talking about some postdoc work I did in Helen Scharfman's lab, and I'm going to be focusing on a unique cell type in the hippocampus known as a Mossy cell. And I'm going to discuss 3 projects, about 3 parts to my talk. They're going to be about 12 to 15 minutes per part. The first one is going to be directly focused on epilepsy because this is an epilepsy seminar, and I'll spend a little bit more time on that talk. However, I'm going to talk about two other parts related to behavior as well as neural circuits of Mossy cells, because these have direct implications for epilepsy. So the take-home message of today's talk, I want you to walk away with three main bullet points. Mossy cells are going to be important in seizures and epilepsy, and I'll present data showing that a normally weak Mossy cell synapse can become pathological in the context of seizures and epilepsy. In addition to this, I'm going to talk about how Mossy cells are important in a variety of cognitive and affective behaviors, specifically behaviors related to learning and memory, tasks that probe novelty, novel locations, as well as novel objects, as well as anxiety-like behaviors, as well as I'm going to emphasize that the Mossy cell circuit is much more complex than we previously thought. Dorsal and ventral Mossy cells differ in their axonal projections throughout the temporal axis of the dentate gyrus. I know a lot of us have been working from home and we haven't had to sit through conference seminars and hear about the hippocampus over and over again lately. So I'm just going to give a really quick overview for those of you that don't work on the structure. The hippocampus is located in the temporal lobes, and it's often cited as a banana-shaped structure. And this is true for both humans and mice. And there's just an example depicted on the right side here. And without getting into too much detail, I want to emphasize that the hippocampus has been very important for learning and memory. And in humans, it's important specifically for declarative types of memories. However, the hippocampus has also been implicated in spatial navigation or spatial coding. There's also evidence that the hippocampus can be involved in affective behaviors, particularly lots of evidence that the ventral portion of the hippocampus can be involved in anxiety or affective type behavior. As well as now some CA2 data showing that it can be involved in social types of behaviors, as well as the hippocampus is known to be implicated in a variety of psychiatric and neurological disorders, one of which being epilepsy. There can be significant structural and functional changes in these conditions. An example of this would be in mesial temporal lobe epilepsy, where you often get mesial temporal lobe sclerosis, where it's this pattern of significant cell loss and damage to the hippocampus. And this is often involved in very severe types of epilepsies where surgical resection may be required. Talking broadly about the hippocampus, I'm now going to focus more specifically on a certain part of it known as the dentate gyrus. And I'm going to talk a lot today in the context of a very simple version of this, the trisynaptic circuit. And this is an example where the dentate gyrus is illustrated in pink here, and it's going to represent the first component of the trisynaptic circuit. And this receives incoming cortical information from such areas such as the entorhinal cortex. And this is going to project to the dentate gyrus. It's going to then propagate to the CA3 and then propagate to the CA1. And within the dentate gyrus, there's going to be three main cells of interest that most researchers study. This is going to be granule cells, which are these tightly clustered cells that are the principal neurons of the dentate gyrus. You're also going to have in the hilus, you're going to have interneurons, which are going to provide strong local inhibition to the dentate, as well as you're going to have Mossy cells, which are in this very unique position to influence dentate activity because they can influence the activity of both granule cells and interneurons. And we think that this is very important because there's a lot of evidence that the dentate is involved in many tasks, and Mossy cells could potentially interact with the other two cells in this area, and therefore have broad effects on dentate-related functions. So as I mentioned, Mossy cells are going to be located in the hilus, and the unique thing about them is there are these large glutamatergic cells in the hilus, and there's no other large glutamatergic cells in the hilus. They're the only main glutamatergic population there. And they have this unique moss-like appearance, which is attributable to their dense clusters of spines known as thorny accrescences. In addition to their morphology, they also have unique, interesting electrophysiological properties where they show high rates of spontaneous activity. They also show low thresholds for activation, which has led some people to propose they might be particularly good at identifying novel inputs or being aroused very easily for kind of new salient information. But also Mossy cells are very susceptible to injury and particularly damage and excitotoxicity. So Mossy cells have been well documented to be implicated in seizures because they often die following seizures. One of the really interesting parts about Mossy cells at a circuit level is that they have complex circuits in the dentate gyrus, which has led to considerable debate about what they do as a net function. Specifically, Mossy cells innervate GABAergic neurons as depicted over here. And those GABAergic interneurons would then in turn activate granule cells. So Mossy cells can provide disynaptic inhibition in the dentate gyrus. However, Mossy cells also can provide monosynaptic excitation of granule cells through a direct connection onto the dendrites of granule cells. In addition to these circuits, we also have these long-range projections of Mossy cells, where an example here would be a ventral Mossy cell can project very long range from a ventral location to a dorsal location on the ipsilateral hemisphere. But there's almost also commissural projections to the right hemisphere. And there's some new evidence that I'll be talking about today that we have to maybe potentially rethink these long-range projections of Mossy cells. And although we've had quite a lot of good quality studies about Mossy cells over the last 20 years, one of the major issues that plagued the field was that there wasn't very good selective methods for targeting Mossy cells. They're particularly difficult to study. They're really not favorable to being worked with or patched, for example. And there wasn't really good methods to target them selectively. But in the late, like 2012, 2015, they started to develop transgenic mice that allowed for very selective targeting of Mossy cells, where we can now use gain or loss of function approaches to study them more directly. And this is important because when I joined the lab in 2016, I thought that there would be three fundamental questions that we should really address in Mossy cells. Specifically, what do they do in epilepsy? There has been considerable debate about whether they're inhibitory or whether they're excitatory. What are their roles and behavior? Specifically, what kind of cognitive tasks as well as anxiety tasks could they be involved in? As well as their role in specific aspects of the anatomy of the hippocampus. Specifically, it's known that there's been some really really amazing circuit studies been done, but a lot of these have been relied on dyes and methods that are just really hard to work with. So now that we have these viral methods where we can do really easy transgenic labeling and viral labeling to identify the circuits, we really wanted to evaluate Mossy cells, and I specifically wanted to look at their axons to see if there'd be potential differences. So the first part of today's talk is I'm going to look at epilepsy, and this is going to be a paper that was published in late 2019 in Cell Reports titled An Excitatory and Epileptogenic Effect of Dentate gyrus Mossy cells in a Mouse Model of Epilepsy. And the reason we wanted to pursue this issue is that there was a lot of debate in the field about whether Mossy cells are excitatory or inhibitory and whether they contribute to epilepsy. One of the main dogmas was that Mossy cells innervate interneurons, as I had mentioned, and those interneurons provide inhibition into the dentate. So when seizures and damage occur, Mossy cells often die. And then there was an idea that there'd be less afferent input onto the interneurons, which would cause local disinhibition and therefore promote seizures. And this is called, this is a theory that was very well cited. However, there's an alternative theory that Mossy cells might become irritable or hyperexcitable. And that was work that was done somewhat in the context of traumatic injury and slices. But there's this idea that Mossy cells actually could also be excitatory in epilepsy. So there was debate about what happens. And the summary of the paper is that we find that under normal physiological conditions, the excitation of granule cells by Mossy cells is relatively weak. But when you start to introduce conditions that are pathological, such as seizures, the excitation of Mossy cells on granule cells becomes very severe, very strong, and so strong that it can contribute to epileptogenesis and seizures. So to address this issue, we did a viral and EEG approach where we did surgery and injected AAV, as well as did an EEG implant. And we focused on a Cre-dependent approach where we used DIR-D2 mice, which is a dopamine D2 receptor mouse that's very selective for Mossy cells in the hilus. And we used inhibitory dreads, which would provide a wide temporal window of inhibition. And specifically what we did is we wanted to try to target as many Mossy cells as possible. So we did a quadruple viral injection approach where we did left and right hippocampus in relatively rostral and somewhat caudal ventral sections. And at the same time, we implanted EEG recordings where we placed over the left frontal cortex, left hippocampus, right occipital cortex with a ground in reference. And the idea here is that we now could, before the seizures, we could pre-inhibit Mossy cells and to see whether they would be making the seizures worse or making the seizures less severe, depending on whether you believed in the inhibition theory or more of the excitation theory. So to do this, we ended up having animals hooked up to the EEG system on day 1 of seizure induction. And we recorded for about 2 consecutive hours just to get really strong baseline EEG signal. We then injected CNO, and then 30 minutes later, we gave pilocarpine. So we pre-inhibited Mossy cells and then evaluated status epilepticus. And just in case anyone asks, we've done a whole bunch of control experiments in supplemental figures where when we looked at control cells versus iDRED expressing Mossy cells, CNO did inhibit those Mossy cells. Also, when we looked at stimulation of extracellular recordings, we could see that inhibiting MOSI cells also had effects at their terminals. So we reduced the EPSP at the dentate gyrus effectively. So we do get inhibitory effects of MOSI cells and their axons. And what we first showed is that inhibiting MOSI cells with inhibitory dreads could attenuate pilocarpine-induced status epilepticus. This is just a representative example of a trace from the left frontal cortex, right occipital, right hippocampus, and left hippocampus. And we can see that there's a really nice and clean baseline period. We then gave a CNO injection. We then gave the pilocarpine injection. And within about 45 minutes, we got very severe seizures that could persist for several hours. This is an hour-long time scale. And we wanted to evaluate whether inhibiting MOSI cells had any effect on status or the onset of epilepsy. And what we found is relative to our Cre-negative group, there was a trend for the first electrographic activity to be delayed, but it was not significant. However, when we looked at the more severe convulsive seizures, we actually noticed that there was about a 20-minute further delay, 15 to 20-minute delay in most of our animals, suggesting that inhibiting MOSI cells actually delayed the onset of seizures. To evaluate this more specifically, we went to the EEG and looked at specific oscillations between 0 Hertz and 100 Hertz, so essentially delta through high gamma. And we effectively found that when we looked at the baseline period, there was no difference in the EEG record between the two groups. However, when we gave CNO during the CNO period, there was a selective increase of EEG power in the theta 4 to 8 Hertz band range. And this was consistent with a paper by Jindi of Kazu Nakazawa's lab that when they did MOSI cell ablation also found a selective effect on theta to be increased. The interesting thing, though, is when the seizures initiated, we actually got the opposing effect. Inhibiting MOSI cells significantly attenuated the EEG power from theta to gamma, so from 4 to 100 Hertz. And although it's hard to tell because this is plotted as absolute, each of these frequencies was reduced by about 30%. And this is illustrated in data that we didn't put in the paper, but overall, there's about a 30% reduction in the entire EEG power. And this can be explained as below, where we have a trace in a representative spectrogram, where once we gave an animal pilocarpine, within about 30 to 45 minutes, they would get SE, and it would ramp up quickly and get very severe and stay severe. Whereas when we gave MOSI cell inhibition, we gave pilocarpine, and when status did happen, it was relatively weak for quite some time. And then as the seizures eventually ramped up, it became more and more generalized. Then we started to notice that it got more severe over time. But it seemed like inhibiting MOSI cells really prevented the seizures from becoming more aggressive at an earlier time point, consistent with what we see with the behavioral data. In our revisions, we had authors, we wanted to address a secondary point of, well, inhibition is only one aspect of this. What happens if we actually look at excitation? So we did the same approach, but we used excitatory dreads, HM3DQ. And the very remarkable thing that we found is that when we excited MOSI cells, the control and the iDread from the previous are just plotted as a percentage now, we saw that seizures happened 50% faster when we excited MOSI cells. And also the convulsive seizures were also considerably quicker. And this is just the same representative trace and timescale and spectrogram on the same timescale as the previous graph. And I've now plotted the vertical lines as the average onset for the different groups. And you can just see that the onset to an electrographic seizure and then shortly thereafter status is considerably faster in exciting MOSI cells. Just anecdotally, in my experience in the Scharfman lab, we've never seen seizures happen that quick. 15 minutes after a pilo injection was insane. Mostly it was 30 to 45 minutes at minimum. So it was just absolutely mind-blowing to me. When we went into looking at specific oscillations, again, we saw a really nice bidirectional effect at the CNO period, where exciting MOSI cells really reduced low frequency power from theta to alpha, whereas inhibiting MOSI cells increased it relative to controls. And then again, we had the opposing effect where inhibiting MOSI cells really reduced power during status, but exciting MOSI cells was roughly equivalent to the control group. To evaluate this in another way, we wanted to look at potentially spikes at the start of status. So to do this, we took EEG traces where we looked at the baseline period and the onset of status. And then I was able to look at the raw traces here. And then using ClampFit, I was able to do a 7 standard deviation threshold and be able to determine how many spikes passed that threshold. And we can see here that there's a lot of spikes and many of them were passing that threshold. When we went to the iDread group, we saw that there were fewer spikes and there were more intervals between those spikes. And this was plotted in summary over here, panel B. There was about a 30% reduction in the number of spikes. However, there was a 30% increase in the interval between spikes, but the overall amplitude of the spikes when they did occur was no different. And this just suggests that inhibiting MOSI cells really reduce the initial drive or the initial excitability of status. Following day, this is 24 hours later, the next morning, the first thing I want to do when I get to the lab is I want to check on my animals because they just had a really tough day with pylo. So I want to look at them. So I turn on all the EEG monitors and I noticed that some animals looked like they were still really having a bad time. There's a lot of ongoing spiking. They looked like disheveled. They did not look like they recovered very well. And then some of the animals had very relatively normal EEG signal for an animal that had recently underwent pylo. And they actually had groomed, they looked great. And I was like, something's going on here. So we needed to evaluate what was happening at day 2. And what I ended up doing is I found that the Cre negatives had lots of persistent ongoing spikes. And when we looked at this more specifically, I was able to actually detect convulsive seizures that were actually quite severe. So this is an example of panel 1 here. There's actually a convulsive seizure. Another example, panel 3, another seizure. And there's also these periods of non-convulsive events that would last for fairly long periods of time. In contrast, when we look at the IDRED animals, some of them spiked, but the majority of them were relatively quiescent compared to the control group. And we again could see some seizures, but they were noticeably less severe when we detected them. And this is summarized in panel B here. There were fewer convulsive seizures, there were fewer non-convulsive seizures, and there were also fewer total seizures overall. And the reason that this is important is the initial insult as well as the secondary insult on the next day could have a lot of effects on neuronal injury. And there's some data that's been provided by that by Bob Sloviter and others. So we were really interested in evaluating this because cell death can happen from hours to days after status. So when we looked at this, we took brains and we took the left hemisphere of a cohort of animals and cut it in the left, in the coronal plane, and we took the right hemisphere and cut it in the horizontal plane because that best evaluated more ventral sections of the hippocampus. And when we did this, we found that when we looked at our Cre negatives, there's no viral expression. And when we looked at adjacent sections, there's considerable damage in the dentate gyrus, the hilus specifically. There's also significant damage in the CA3, and then there was significant damage in the CA1. When we look at an IDRED animal, which still has really beautiful viral expression after 3 days, we see nice viral expression, but in adjacent sections, there's almost no fluorojade damage. Specifically, the hilus seemed very well preserved, and the downstream target, the CA3, was also very well preserved. However, the CA1, which could have inputs from other regions, was actually equivalently damaged. And this is plotted on the right here. So the hilus had about 50% less cell damage, as did the CA3, actually even more in the CA3. And then when we looked at the CA1 and GCL, which are areas, the CA1 was equivalently damaged, and then the GCL was an area where there's almost minimal damage as granule cells are relatively resilient to pilocarpine-induced damage. To get at this more specifically, were these MOSI cells, because there can be, as the reviewer pointed out, large cells in the hilus aren't always MOSI cells. They could be somatostatin interneurons, there could be other things. So we ended up looking at GLU2R3, which is a MOSI cell specific marker. And in our control animals, we actually noticed that there was considerable cell death or cell loss in these animals because the hilus was relatively sparsely labeled with GLU2R3. And this is depicted both in dorsal sections as well as ventral sections. But in our inhibitory dread animals, we noticed that there was actually pretty good MOSI cell preservation, both in dorsal and ventral sections. And this is plotted just down here where we see very nice protection. However, when we looked at other cell types, notice that the y-axis is half here. We noticed that there was actually considerable loss of somatostatin neurons. There was like as few as 5 cells per section in some of the animals in the dorsal sections. So somatostatin seemed vulnerable, but inhibiting MOSI cells protected MOSI cells specifically. Finally, we wanted to evaluate chronic epilepsy because this is what we really wanted to determine whether inhibiting MOSI cells had like long-term functional consequences. So we ended up doing the same approach, but instead of sacking the animals at 72 hours after pylo, we let them recover for 4 weeks, and then we monitored epilepsy for 2 consecutive weeks with 24/7 video EEG. And what we looked at was the total number of seizures, seizure frequency, days with a seizure, consecutive days with a seizure, which we could kind of infer as clustering-like behavior, as well as the duration of each of these seizures. And what we found here is that the Cre negatives had really severe seizures that could last up to a minute, and they were very frequent. In contrast, we noticed that the IDRED animals had fewer seizures, and when they did have seizures, they were generally less severe. And this is plotted just through those panels. Overall, inhibiting MOSI cells at the time of status had all these long-term benefits on subsequent seizures. So at this time point, we did not use CNO. This was just, CNO was only at the time of induction. So this is just, if we inhibited MOSI cells during the initial period, we had long-term benefits in chronic epilepsy. Finally, to get at circuit mechanisms, we ended up going to slice physiology to evaluate, well, how can Mossy cells be excitatory? And we used an optogenetic approach where we injected virus into the hilus, and then using distal slices, we were able to focus on Mossy cell axons in the molecular layer while patching individual granule cells, and we could determine whether Mossy cell activating Mossy cell axons could excite granule cells. And what we found is that we would make slices, we would prepare slices, and then we record under normal conditions. And then under normal conditions, we effectively found that the light response of 2 milliseconds was always really incredibly small. There was a very weak excitatory EPSP at best. But what we could do is when we started to simulate seizure-like conditions, we started to notice that the same cell started to grow in its response. And over about 20 to 30 minutes, the same light pulse in the same cell, which basically was minimal before, could now evoke multiple discharges in granule cells that could last for more than a few seconds, which is equivalent to a paroxysmal depolarization shift. So this was a really surprising finding to us. However, an individual cell is not going to be representative of an epilepsy network disorder. So we wanted to evaluate, well, what's the overall effect on the dentate? And to address this, we went into interface recordings to do extracellular interface chamber to do extracellular recordings. And when we did this, we ended up doing the same approach where we did surgery, we made slices, we started to record, and then we started to simulate status. And when we got to the status time point, we were recorded in multiple layers. We recorded in the intermolecular layer, downstream in the CA3, downstream in the CA1, and then downstream in the subiculum. And this kind of addresses that trisynaptic circuit I talked about at the start of my presentation. And when we did this, the IML light response was immediate. And then downstream in the CA3, it happened at a delay, but we had noticed epileptiform-like activity in the CA3, which continued into the CA1 and then continued into the subiculum. And this just shows the delayed response from IML, CA3, CA1 subiculum, showing there's a nice temporal gradient suggesting that it's propagating down the trisynaptic circuit. But to address the trisynaptic circuits directly, we use this drug called DCG4, which is going to inhibit the axons of Mossy cell or Mossy fibers, which are the granule cell axons. So if we inhibit the granule cell axons, you should presumably prevent the downstream events if it's granule cell mediated. And that's exactly what we found. When we were able to record downstream events in CA3 and subiculum, we blocked the granule cell axons, it completely blocked this effect. And then when we washed it out the drug, it came back. So this suggests that the epileptiform activity was mediated through granule cells, propagating, discharging and propagating throughout the hippocampus. So in summary of part one, we found that inhibiting Mossy cells during status conferred numerous beneficial effects. It delayed the onset of seizures. It also reduced the EEG spikes and power during the onset of status. It also reduced day 2 acute seizure burden, as well as when we looked in the cohort of animals at chronic epilepsy, it also reduced their burden. Furthermore, it also reduced neuronal injury, and this appeared to be selective to Mossy cells and CA3 pyramidal neurons because CA1 was unaffected. And then finally, exciting Mossy cells or Mossy cell axons during status or simulated status in slices could produce adverse effects. Specifically, it accelerated seizure onset. It could detonate granule cells individually when we did optogenetic experiments in slices, as well as it could generate epileptiform events that propagated throughout the hippocampus when we looked at extracellular recordings. So the next part of today's talk is going to be a behavior section. And when we started this work, there was relatively few studies on Mossy cells and behavior using selective techniques. There's been several nice labs that have been doing work such as Kazuo Nakazawa, Ivan Schultes, Jim Neerum, Yuri Bijaki, Attila Lazanzi. And collectively, some of these papers were starting to provide evidence that Mossy cells could be involved in spatial coding or navigation, novelty or novelty detection, interaction of novel objects. But also less was considerably known about anxiety. And unfortunately, although some of these studies were really well done, they were very focused in specific questions. And we were kind of interested in a more broad approach of, well, if we evaluate multiple behaviors, which ones are going to be most promising if we want to design these projects in the context of epilepsy, for example. And the other issue that we wanted to address is that historically, a lot of the studies on Mossy cells had been mostly done in males, or when they did use females, they were often pooled. So there wasn't a clear view of sex differences, and we wanted to address this. The one thing I wanted to say is that I actually got really good news this week. Just as of Monday, the paper was accepted for publication in the Journal of Neuroscience. So if you're interested in this, I'm only showing a part of the story today, but keep an eye out for that. It should be out soon. Next, what we'll do is I'll just give an overview of the experimental design, and it's similar to what we did in the previous study, where we used these DR2 mice, and we did Cre-dependent viral targeting of MOSI cells. We specifically used excitatory dreads as well as inhibitory dreads for gain of function and loss of function. And we did the same approach where we did 4 viral injections into the hippocampus. This time we did a little bit more caudal and a little bit less ventral, just because it's harder to target ventral reliably. And overall, what we found is that in the control, you really don't see anything. But when we go to a dread animal, we see that there's considerable viral expression from rostral all the way to caudal sections. And we were able to quantify this. So we counted individual M-cherry cells in E-dread or I-dread mice. And we found that there was about 20 plus cells per section throughout the entire, all the sections we analyzed. Furthermore, we also could notice that the axon was equivalently bright when we made these grayscales and we just looked at their difference from background. We noticed that we pooled E-dread and I-dread, and we found that essentially all of them had a similar brightness of their axons. So by doing this approach, we thought that inhibiting or exciting MOSI cells could have effects on either the direct or the indirect pathway of the MOSI cell circuit. So specifically by activating or inhibiting MOSI cells, you could affect the drive of MOSI cells onto granule cells directly. Alternatively, you could also have indirect effects where by activating or inhibiting MOSI cells, you could have effects of MOSI cells on the interneurons, which then in turn inhibit the granule cells. So we weren't really sure what exactly effects we could get, how this could affect cognition. So we wanted to do a variety of cognitive tests to evaluate this. The first thing that we ended up looking at is contextual fear memory. And this is a task where we just place an animal or a mouse into an operant chamber, and we would administer 3 shocks at 0.5 milliamp. We would do the shocks as a 1-minute interval, and we would give CNO 30 minutes before training. And what we found is that the controls are depicted in blue, the I-dreads are depicted in red, and the E-dreads are depicted in green. And what we found is that at the baseline period B1 and B2, there's really low baseline freezing. But what you see is this over time from shock 1, shock 2, shock 3, there's this nice ramp up of a learning curve, specifically in the I-dread and the control mice. The E-dreads showed a delayed learning curve, although they did show more freezing over time, it wasn't as sensitive to this. And this is demonstrated in panel C, the overall average, where there was about a 50% reduction in the amount of freezing in E-dread animals or exciting MOSI cells relative to controls and inhibiting MOSI cells. The notable thing about this is that when we separated it out by sex, we found that males actually had the same trend as females, but they froze less overall. And therefore, the females really were the primary drivers of this behavioral phenotype. 24 hours later, we placed the animals back into the original context. This time, no shocks, no CNO. And we just wanted to evaluate, well, how well do they remember the previous day? And we find that the control and the I-dread showed equivalent freezing throughout the entire task, whereas the E-dreads once again showed about a 50% of the amount of freezing. And this is again depicted as the overall average on panel G. And then when we went to the sex differences again, we noticed that a similar trend occurred in both females and males, but it was again more pronounced in the female cohort. Next, we wanted to evaluate novel object recognition because there's been some papers and some recent evidence that MOSI cells might be important for novelty detection or novel objects or interacting with novel objects. So we did a task where we placed 2 objects into an environment and we look at how MOSI cells could influence this. So we placed 2 identical objects, A and B, and we allowed the mice to explore. And we inhibited MOSI cells or excited MOSI cells 30 minutes before we did this. And what we measured is something called a discrimination index, which is essentially your preference for B over A or A over B, depending on how you do it. And the idea here is that if there's no preference, your score should be close to 0. And that's indeed what we found on training. All the bar graphs showed that there was really no preference for either object because they're identical. Same thing when we separated out by sex. And then when we looked at the total time exploring, there was also no difference between any of the animals. However, when we tested 1 hour later, we replaced object B with a brand new object. So the idea here is that mice should have a preference for this new object and want to interact with it more than an object they haven't, that they're familiar with. And when we did this, we found that inhibiting MOSI cells really promoted a positive DI score, meaning that they have a strong preference for object B over object A. But when you look at the E-dread animals, they really, again, show basically a flat score. It's around 0. When we looked at sexes, though, we noticed that this effect was most prominent in the male mice, and the females actually didn't show this phenotype. And this was important because the overall exploration was the same. It's just that exciting MOSI cells in males really didn't motivate them to explore a new object. To continue this theme of MOSI cells and novelty, we ended up looking at how MOSI cells could be involved in novelty suppressed feeding. There was a really nice neuron paper by Estefania Azevedo that showed that D2 neurons, including MOSI cells, could be involved in eating and feeding behavior. So we wanted to address this in this work as well, using gain and loss of function. So this approach is where we food deprived a mouse for 24 hours, water deprived it for 2 hours, and then place it in a very well-lit novel environment and place food in the center of the arena. And this task is thought to challenge their hunger versus their innate aversion towards like an open field where it could be potentially dangerous. And when we did this, we found that inhibiting MOSI cells reduced freezing or reduced the latency to eat, significantly relative to the E-dread group. So it took about 4 minutes to eat, 4.5 minutes to eat if you were an I-dread mouse, but it took upwards of 6 to 7 minutes, if not longer, if you were an E-dread mouse. So it just facilitated the latency, or it facilitated the time to eat. And again, we found that although there were, comparable trends in males and females, again, this effect was most robust in the female cohort, which is also notable. And then finally, one thing that we wanted to do is again, continue this theme of novelty, but we looked at objects in the home cage. So unlike all the other tasks where we, move the mice to a new room and we're doing all these procedures that could have some innate anxiety and testing stress by itself, we wanted to do a task where we try to keep the mice as minimally as aroused as possible and just introduce something that allows them to explore if they want or not explore. And this is a task that was developed by Hannah Bernstein and Helen Sharfman's lab. And we published a paper, I think the year before this, in neuroplasticity, and we wanted to kind of use this paradigm using gain and loss of function. And the really interesting thing that we found is that when we excited MOSI cells, they really reduced their exploration of these objects relative to a control. And then when we inhibited MOSI cells, we actually facilitated their exploration. And this was seen as an overall trend in both male and female mice. And then when we did minute by minute analysis, the same effect held. And you can really see that it was really consistent exploration relative to the E-dreads. It just completely didn't go all over the place. It was very stable. The notable thing though, is we wanted to evaluate how inhibiting or exciting MOSI cells could affect the circuit. So we ended up 90 minutes after this task, euthanizing the mice to evaluate how dread effects could affect the DG circuit using CFOS. And unsurprisingly, when we did this, we looked at CFOS with excitatory dreads, and we found a lot of CFOS in the hylus consistent with the E-dreads exciting Hyler cells, or MOSI cells specifically. The surprising finding, though, is when we looked at I-dreads, there was very sparse activity in the hylus, but we ended up getting a lot of granule cell activity, specifically about twice as much granule cell activity. So this suggests that inhibiting MOSI cells actually starts to promote the recruitment of granule cells, whereas activating MOSI cells just seems to really activate Hyler neurons. And we saw this both in coronal sections as well as horizontal sections. So the summary of this talk, of this paper, was that MOSI cells were important for several cognitive and anxiety-like type behaviors. We found effects in contextual fear conditioning, effects in novel object recognition, effects in novelty suppressed feeding, novel object exploration, and there's several others that I didn't talk about. So keep an eye out for that new paper that will be coming out hopefully soon if you're interested in learning more. I also want to emphasize that sex mattered. Males and females often differed on some of these tasks. So I think that when you do your experiments, you really want to consider adding males and females because you might be underestimating your effects if you only study one sex. And then finally, as I just mentioned with CFOS data, exciting versus inhibiting MOSI cells differentially modified the dentate circuit. Exciting MOSI cells increased Hyler phos, whereas inhibiting MOSI cells increased granule cell phos. So there's going to be effects on these indirect versus direct pathways I talked about in that circuit schematic. And then finally, I'm going to talk about a paper that I'm really excited about. It's really near and dear to my heart. And this is addressing the anatomy of mossy cells. This is a paper that is now under minor revision. It's been resubmitted and it's currently under review. So I hope we get good news in the coming days, but if you follow me on Twitter, you'll find out soon enough. This paper talked about how dorsal and ventral mossy cells may differ in their axonal projections throughout the dentate gyrus of the mouse hippocampus. So what I wanted to do with this paper is we've been working with mossy cells for quite some time, but previously most of the work I had done was looking at multiple injections, a quadruple viral injection approach. And I really wanted to say, well, can we evaluate this more carefully where we just look at dorsal versus ventral and look at whether there's different projections between these two. And the reason for this is that we already know that there are some potential dorsal ventral differences that may exist. There's been papers previously suggesting that calretinin can be expressed in ventral mossy cells of the mouse, but not dorsal mossy cells. There's also electrophysiological evidence showing that ventral mossy cells might actually show more bursts in a slice relative to dorsal mossy cells in response to pharmacological agents. So there's evidence that these might differ, but no one's really addressed this systematically. And because we already had the mossy cell specific transgenic mice and viral approaches working in the lab, I thought this is a really great project to address mossy cells and mossy cell axons. And one thing that we were able to do is we were able to take measurements of the molecular layer to quantify the mossy cell axon. So the molecular layer can be defined as bordering between the granule cell molecular layer border, which is depicted in this red dash here, and it will extend out into the fissure. And that's the total molecular layer, but it can be divided into thirds, which is the inner, the middle, and the outer. And historically, mossy cells were thought to reside in the inner molecular layer only. Furthermore, we were interested in studying potential regional differences where there's an upper blade, a crest, and a lower blade of the dentate gyrus, because there's been several electrophysiological papers suggesting there might be upper versus lower differences in dentate function by itself. And we were curious whether this would also be true for mossy cell expression. So to address this issue, we ended up doing viral injections in dorsal mossy cells and then just evaluating their axons throughout the central temporal axis. So here's an example where we did a single viral injection into the left hilus, and then we were able to evaluate just section and look at sections all the way to the caudal and more ventral portions of that hemisphere. And this is where we did coronal sectioning. And this is just a representative somatic of more rostral to more caudal sections. And what we see in these more rostral sections, we have lots of cells near the injection site, and there's a green axon that's relatively faint, but it's bordering the granule cell IML border. And as we start to go towards more caudal sections, starting in C5, C6, C7, the cell number starts to drop off, but we start to notice a more prominent width of the axon. Specifically, you see this kind of specific tight band here, but then it starts to extend further into the molecular layer. And I wanted to evaluate this more, so we kept going more caudal, and then we also started looking more ventral. And when we did this, we could look at the upper blade and the crest, and you can see that this is a really wide axon plexus now. It seems to definitely extend beyond the inner molecular layer. But as we went towards the more ventral portions of these sections, we noticed that it started to get thin again, and then it actually started to separate away from the granule cell IML border. And this actually starts to project primarily to the middle molecular layer, and I'll show quantification for that shortly. However, we wanted to evaluate commissural projections as well. So we took the other hemisphere and cut it in the horizontal plane because that's going to allow us to best evaluate dorsal ventral. Particularly the ventral hippocampus is really hard to evaluate in coronal sections. It gets quite messy, whereas horizontal sections are fantastic for evaluating this. So what we found is when we looked at relatively dorsal horizontal sections towards more mid and towards more ventral, we found the same effect that in these sections, there was a significant molecular layer labeling, and it seemed to be quite wide throughout the molecular layer. And then as it became more ventral, it started to shrink in its total width, and it started to get further and further away from the GCL IML border. The other notable thing that we saw was that there was significant cell labeling in the non-injected hemisphere. And almost in all sections we evaluated, we saw a few scattered cells. The cool thing is when we actually went and did high resolution stacks and we could look at calretinin or GLUT2R3, these were mossy cells because they had the physiological or the morphological appearance, but also the immunohistochemical labeling. So this is a really interesting approach that might be to anterograde or retrograde trafficking of mossy cells using these viral approaches. The thing is, though, when we look at this, dorsal mossy cells aren't really thought to do that. Like historically, there was no basis for this. So we were a little bit worried about, is this contamination? Is there some weird thing going on with our virus? Like, how can we explain this? So we wanted to evaluate, well, if we look at ventral mossy cells, what happens? Do we see the same weird axon plexus? And the good news is when we injected really ventral sections, we got a whole bunch of ventral mossy cells. And then when we looked throughout the entire septotemporal axis, all of the axon was always adjacent to the GCL IML border and always very consistent and tight. And clearly, as we went further and further away from the injection site, there's no cells present. And then when we go into the contralateral hemisphere to look at commissural projections in the contralateral hemisphere, the right side, we did this horizontally. We see the same effect, that this was a ventral injected animal, but we see this nice consistent trend of axon labeling along the GCL IML border for all sections. The notable thing, though, is that historically mossy cells were thought to be, the commissural projections were thought to be homotopic, meaning the left ventral hippocampus would project to the right ventral hippocampus. But when we did this, a left ventral hippocampal injection projected all the way to the tip of the right dorsal hippocampus. So their axon would be heterotopic. And we saw this both in dorsal and ventral injected animals. So we have all this nice descriptive data. We needed to quantify it. So we ended up taking horizontal slices where we could evaluate the GFP signal as a percentage of the molecular layer in dorsal and ventral sections. And the way we came up with doing this is we looked at a measurement called inner GFP, which is the distance between the GCL IML border and the most stable or consistent band of the starting point of the GFP axon. We also were able to look at this relative to the outer point of GFP axon. And then we did the difference as the total GFP width. There was sometimes where you get these individual boutons, but I mean, one is not gonna, it's gonna cause way too much variance. So we looked at just like, where's the most consistent, like draw a perpendicular line, and can we kind of follow the axon as like the majority of the bulk of the axon. And we were able to do these measurements, and because we've now measured the molecular layer width, we can separate it into thirds. So 33% threshold would be IML, 66% threshold would be MML or IML to MML, and anything beyond the 66% threshold would be OML. And when we did this, we found that the dorsal injection resulted in dorsal sections, resulted in an axon that always was below the IML threshold. So it was always within 1 or 2% of the GCL IML border. But when we looked at where the GFP started in those ventral sections, they almost always began in the MMLs, meaning they were around 35% or so. So they began in the middle molecular layer. When we looked at the outermost part of the signal, we found that almost all of these ended up in the MML to OML border, because most of them averaged around 67 to 69%, which was really surprising. And then finally, we were able to look at the total width of the axon. And we see that, again, IML is in a dorsal injection, it's from 0% to about 67%, meaning that it's about the 2/3 of the molecular layer. Whereas when we look at the ventral injection, it's below the 33% threshold. And because it primarily starts at 33% over here, it basically means it starts and primarily ends in the middle molecular layer. When we did the ventral version of this, it was very much consistent with what's been predicted in the literature previously. Everything basically was below the 33% threshold. So all the axons began in the GCL IML border. They all terminated within the IML GCL border or GCL IML border. And none of them extended beyond that. So ventrals were very consistent with what's been known. The dorsals were the surprising effect. And we can show this by mapping this by looking at dorsal versus ventral directly. So when we look at a dorsal injection mouse in blue and ventral injection, although there's different dorsal ventral differences in a dorsal mouse, we see that overall they're fundamentally different than a ventral injected mouse for the inner distance. The same thing happens for the outer distance, and the same thing happens for the total width of the axon. So there's clear dorsal ventral differences in dorsal and ventral mossy cells. And just to illustrate this point further, we could do a dual viral injection approach where we inject the dorsal hippocampus with M. cherry and we inject the ventral hippocampus with YFP. And what we can see here is that the M. cherry begins in the IML and there's some yellow kind of overlap of virus, but it extends further into the molecular layer throughout the entire sections. But as it progresses from more dorsal to more ventral, the proximity to the GCL IML border gets further and further away, and it starts to primarily terminate in the MML. Whereas these ventral set injected animals with YFP, they're primarily restricted to the IML border for all sections. So there's this clear dorsal ventral difference. And the way that we can explain this is that calretinin labels ventral but not dorsal Mossy cells. I think the reason that no one's really gone on to the fact that dorsal Mossy cells may differ is there's no good antibodies for dorsal Mossy cells. So when we do this approach, we can do calretinin in red, we can do a dorsal injection, and we can see that all of these green Mossy cells don't have any calretin soma labeling. And even the axons that we see in the IML really don't colocalize, they almost seem distinct. And when we go into these more ventral sections of the same animal, we actually can see that there's nice cell somatic labeling in the ventral hilus and in its IML, but we see this GFP axon that's basically in the middle molecular layer adjacent to it. So there's this clear separation. When we did the ventral injections though, GFP would be broadly expressed in the hilus and the IML, and the same thing with the same pattern as the calretinin, where there's a lot of yellow in the ventral. And then when we go into dorsal sections, we see no cells in the hilus, but we see very strong colocalization in the IML of these animals, suggesting that calretinin is only labeling ventral Mossy cells. And this is also in these in the right hemisphere of these animals, we're able to then do the same approach where we look at calretinin and there's this very clear separation of calretinin in red and the GFP signal in these ventral sections. But when we look at the contralateral hemisphere, there's just a bright yellow axon in the IML, suggesting that dorsal and ventral Mossy cells have different termination zones. So the summary of this, and I'm almost finished, thank you for your attention, is the Mossy cell circuit may need to be reevaluated. Specifically, this distinct projection to Mossy cell should be considered when evaluating dorsal ventral differences in dentate function. If we look at the dorsal hippocampus or the ventral hippocampus, we can look at these ventral Mossy cells in green here, and we basically say that it's very reliable. It always projects to the IML. However, when we look at dorsal Mossy cells, it starts to get confusing. Basically, dorsal Mossy cells are going to innervate the IML of dorsal sections, but they can also extend into the IML or to the MML of dorsal sections. But when you look at dorsal Mossy cells in the ventral hippocampus, these axons primarily begin and end in the middle molecular layer. So this suggests that dorsal Mossy cells might have different inputs or circuits they could interact with. One that comes to mind immediately is medial entorhinal cortex, where there'd be perforant path inputs into the middle molecular layer. Another interesting possibility is that there's some nice evidence that Mossy cells provide some of the first glutamatergic inputs into adult-born neurons. And because adult-born neurons primarily have dendrites that begin in the IML as they're maturing, it might be that ventral Mossy cells are primarily the innervation of adult-born neurons, and maybe dorsal Mossy cells might activate more mature granule cells, for example. So this could have very interesting implications about how dorsal and ventral Mossy cells affect the dentate circuitry differently. And then finally, just in closing, I have an absolutely fantastic lab in New York. Helen Scharfman was an absolute fantastic mentor that spent a lot of time with me going through all of these projects, lots of feedback. Dr. Yiling Liu was really just essential in doing some of the electrophysiology work for the cell reports paper. Vinod Yaragurdi was really important for the behavior work. David Alcantara, Hannah Bernstein, John LaFrancois, and others in the lab were also just fantastic colleagues. NYU, NKI were really great. And then also I had postdoctoral support from AES and Cirque Savoy Foundation, and Helen had a grant from NIH. So in that, I can close with questions. And if you don't like questions or get nervous, you can email me or message me on Twitter. Thank you very much, everyone.
Justin, that was a great job. That was a great talk. Your anatomy looks really amazing. So we have time for questions.
I have one.
Type it in. Oh, Tally has a question.
Just one second, I lost my headphones.
So first of all, really a tour de force. I mean, boy, you did well in New York. This was really fun. I have a question that pertains to your project 2 and project 3. I mean, project 1 is so clear there's just nothing going. So maybe I'll start with the second question. You have shown us a variety of behavioral consequences of manipulating the Mossy cells, but as far as I can tell, you manipulated all of them. And then in project 3, you show this amazingly different connectivities of the dorsal versus ventral. So it'd be kind of fun to speculate, you know, do you think that the different behaviors or types of learning or whatever that you found in project 2 perhaps pertain to different subpopulations or anatomical distribution of the Mossy cells, dorsal versus ventral? But before you answer, let me ask the second question because they maybe pertain, maybe related. So you've shown us a lot of behaviors that are influenced by manipulating the mossy cells, but you haven't shown us any behavior that are not influenced by manipulating the mossy cells. I mean, do you think you may be simply doing something to the state of arousal or attention or the ability to focus on training and learning rather than actual circuitry of learning? So these are the two questions. And
sure. Because it's a short talk and I had a lot more slides, we actually do have some tasks that did not have behavioral effects. So I just picked the highlights. We actually were really shocked, but we did novel object location, mostly based off the fact that Ivan Schultes has a really robust effect on that. And we did it and we found no effect. And I show that as the fourth figure in my J Neuroscience submission. And I think part of the reason for that is that it could be that the way we did a short interval of 1 hour where they did an over day, like they did a 24-hour interval between learning and testing. So, or training and testing. So that could be an effect. It also could be like we had a relatively far width between the location of the two. So we may have rendered the task less dentate dependent because I think they had a really close proximity for location, whereas ours was like double or triple the width. So I think the dentate can be sensitive to certain types of tasks, but I didn't show the ones where we didn't have that great of data. We also looked at things like open field behavior, and there were some trends, but there wasn't anything overly compelling. So, you know, there was some data that wasn't affected. So I don't necessarily know if everything is going to be specific to mossy cells or mossy cells having broader effects on the dentate and having some effects on arousal, but we didn't see any weird stereotypic behavior or anything that would suggest that, you know, the mice were abnormal per se in the majority of studies. I just think that there might be some tasks that were more or less sensitive to those behaviors. Regarding your first question about dorsal ventral, I think that's fantastic. And it was something that in hindsight, as we were we started the behavior paper about a year before we started the anatomy paper. And the anatomy paper was supposed to be this little simple thing that kind of morphed into this bigger thing. And then we had COVID and a lot of the stuff I had planned to do kind of got thrown to the wayside because we got shut down being in New York early in spring. But I think what we ended up doing is there are now some papers that have actually shown like ventral mossy cells specifically are important for novelty. There was a really nice paper in Current Biology recently by Fredes. And there's also been some recent papers by Young-Sik Oh's group that actually have found a similar finding of dorsal ventral differences in mossy cells. And it's actually a preprint right now. I think it got sent to one of the Nature groups. And they actually showed there's all these transcriptional differences that differ, but there's also behavioral differences. So I feel like that's an issue that I wanted to pursue, but by the time we kind of were getting all these pulled together, we kind of had missed the boat a little bit. Other groups started to pick up on these issues. So it's something that in hindsight, I wish I had done more of though.
Can you,
thank you.
This is Chuck Reback.
Hi, Chuck.
Can I talk? I'd like to ask you about the fluoro-jade data in the first part of your talk.
Sure.
Can you explain why the mossy cells are not dying off in the eye dread?
That's a tough question.
And can you comment on the role that excitotoxicity plays in the regular normal situation following pilocarpine?
Sure. So we were not too sure what we would get out of the fluoro-jade data when we first did it. It was kind of an optimistic, well, maybe if the seizures are less severe, maybe there'd be less excitotoxicity. Potentially, you know, just by having mossy cells fire less, there might be less, you know, calcium buildup. Essentially, like Helen's done some work previously showing that calcium chelators could essentially block some of the excitotoxicity in mossy cells. So by having inhibiting mossy cells or reducing their activity, that might prevent some of the overwhelming depolarizations of mossy cells, which could contribute to their excitotoxicity. And then, you know, if we're effectively also reducing, like looking at some of the spike data, for example, we see that there's just less initial activity in the dentate. So if we're preserving some of the detonation of granule cells, that could potentially also have downstream effects on CA3. But I mean, it's speculative at best. I mean, I really wish I could have a better answer for that, but it's a challenging issue. The interesting thing was CA1 was not affected, and that could be due to like temporal mnemonic inputs and other inputs, but it did seem that like local dentate circuit was relatively preserved. potentially just by having them, detonate less. Whether there's depolarization block and other factors go in, we're not too sure. It's an interesting issue. And then your second point, sorry, I maybe forgot that already.
I was just interested in knowing about the connections between granule cells and mossy cells, whether or not you're blocking those at all with the iDREAD, or whether it's a direct effect right on mossy cells.
I think what we can get is like the terminals, like we've done some electrophysiological logical things like extracellularly, where if we inhibited mossy cells and we stimulated, we would get reduced extracellular recordings. So we think that there's less interactions between the synapse, like at the terminal level. So since mossy cells interact with granule cell mossy fibers as well as interneurons, we think that there's probably somatic effects, but we also had this gut feeling in the lab that we thought for a long time a lot of the effect was at the axon, especially in the context of status, because it's going to be a glutamatergic synapse that could basically dump large amounts of glutamate, which could further contribute to excitotoxicity. And that was one of the reasons when we did a lot of the electrophysiological data and opto experiments, we basically focused exclusively on the axons. A lot of the groups that have done previous work on mossy cells have really looked locally, but the local interactions of mossy cells might be actually different than their longer range projections, because the local mossy cell, about 25% of the axon is in the hilus, and that could have an effect on granule cells, granule cell mossy fibers, but also a whole bunch of interneuron axons as well. Whereas the IML, or the the molecular layer axon, although there is some innervation of interneurons, it's overwhelmingly more monosynaptic of mossy cell to granule cell. So I think there is like local circuit effects between the two molecular layer inputs, hilar inputs, and we think that inhibiting mossy cells have somatic effects, but also terminal effects that could be important for that toxicity.
Well, congratulations on a wonderful group of studies, and give my best to Helen, please.
Thank you very much.
Okay, so we have a couple of message chat questions, and then Laura, you have your hand up. So I'll do Jessica Chansey first, and then Laura. So Jessica says, great talk. Have you guys looked at whether or not dorsal versus ventral mossy cells differentially innervate different cell types, interneurons versus granule cells, adult-born versus early-born granule cells?
So that was something I tried to address at the end of that circuit diagram. Basically, no, it's a really great point. That was something that I wanted to do. I've kind of done, I'm gonna flip through like 100 slides, I'm sorry. I've done some GABA stuff that we showed in supplemental. So we have looked at VGAT as well as GABAergic markers. And when we've done this, we really didn't see any convincing co-localization. However, there's just a dense red VGAT or a dense red GAD67. So whether these are juxtaposed and have influence where they have different, like we didn't do a lot of co-localization with pre-post synaptic, but we didn't see compelling evidence that mossy cells would innervate GABAergic neurons or granule cells more than the other. We have to still look at that. But this is something that we wanted to pursue, but then the lab got shut down. So we kind of started to submit papers instead, and I eventually moved on from that work. Regarding the second point about adult-born versus immature neurons, that's just something that we started to dream up I started to dream up when we started writing the paper, and I started noticing that, well, if you have an IML versus an MML, there could be potential different segments of like the more proximal dendrite versus the more distal dendrite of a granule cell. And I kind of phrase that in the context of immature versus mature granule cell, but more work could definitely be done there.
Okay, we're gonna go Laura, you're up, and then I'll address Momoko.
Hey, Justin.
Hey.
So much data, my head is spinning, but it's all awesome. My question is about way back at the beginning of well, not the beginning of the first talk, but the end of the first talk, when you were looking at the intracellular recordings in granule cells, when you were optogenetically exciting the mossy cell axons.
Sure.
That's so interesting that like individual stimuli give these tiny little excitation responses, but then if you stimulate with trains or repetitively, you kind of ungate this like monster input.
These are single pulses. Just we would wait at least 1 minute, and it was always a 2 millisecond pulse. So we didn't do trains in this, but we have done trains in a different preprint I'm on.
So it's that you gave single pulses, but it was later that you would
yeah. So
start.
like we would, the way we would design it is we'd basically like have the granule cell patched, and we'd have like we get a light response, and then we'd set it to like record every minute. And then like after about 10 minutes or 5 minutes, once we got like nice baseline consistency, we'd then wash in the seizure cocktail. And then over the next like 15, 20 traces, we'd just start to see this monster response, but we would never like do trains or repetitive. These were just one-off.
This is, you think, because of the seizure cocktail?
Yeah, well, I mean, we're doing things like we're using GABA drugs, we're essentially increasing potassium, and these are all things that are based off the fact that these events kind of happen during status. So we could have done like a pilo drug per se, but we just simulated it in vitro to the best of our ability. The thing is we've also, like, through the reviews, there was pretty extensive questions about this, like our model. So we've done these independently, and what we find is that just increasing potassium, just reducing magnesium, just using GABA drugs, all of those increase this effect. It's just they're additive. And because most of these happen in parallel in status, we kind of just did it all at once. So we just see that these can become these really prominent events. And like every time we did this, we always got the same result. So it's very consistent. And we've also done in our preprint, there's like just a dedicated EPHIS preprint. We show that there's some effects where it depends on the timing. So like you can also stim perforant path, and whether you activate MOSI cells before or after, that can cause inhibition or excitation. So there can be facilitation or depression depending on when inputs happen, but under a seizure, like under normal physiological conditions. But under seizure conditions, a lot has changed and it's just more excitatory. But there was a selective increase because when we looked at other weak synapses, MOSI cells were, 10-fold higher relative to their baseline, whereas other synapses like the CA3 back projection was like 2 times higher. So it was a very specific enhancement of the MOSI cell synapse, although it does take it with a grain of salt that all synapses should be more excitable under a seizure cocktail.
Okay, thank you. I was misunderstanding. I was thinking that you were getting this through some kind of plasticity mechanism or something.
No, we tried to avoid that. There is definitely like an LTP BDNF dependent mechanism on that. But we didn't want to do any like trains or repeated pulses stuff. So we just did single one-offs with significant delays.
And do you have any plans to follow up these anatomical studies with exploring whether there's some kind of co-plasticity between MPP and dorsal MOSI cell ventral projections?
I don't want to say too much. I'm not in the lab anymore, so I'm in Toronto now. So I would love to do this in my own lab, but I also think Helen wants to address some of these issues. So we're in that position where we're in discussion, and I don't want to say too much about what her plans are because she's dreaming some ideas up. But I definitely want to pursue some of these issues if I ever get my own lab. So
cool. Thank you.
Yeah, thank you.
Okay, so we have a question from Momoko. So Momoko says, very beautiful talk, thank you. It is interesting that MOSI cell has differential effects on cognitive versus anxiety-like behaviors by gender. I think sex differences. Since you looked into anatomical projections, did you see any sex differences in anatomy? Any hints why this is different?
Very good question. Again, I cherry picked some of the data because we've done some anxiety stuff where we did see effects in males and not females, for example. So I just presented the data that I could fit into the talk in a relatively short manner. When we've done this, we actually did do anatomical measurements of blue and red. Blue would be males and red would be females here. And when we looked at just the overall like location of everything, whether it's also a D2 mouse or a CRL mouse, which is an alternative MOSI cell mouse, we tried to split it about 50-50. There's no difference. Anatomically speaking, the axon projections of dorsal and ventral are both the same, whether we do dorsal sections or ventral sections. Actually, most of my, well, about 50% of quantification was in females, but most of my representative images are actually from female mice. I didn't even notice that until a reviewer asked us to say, specify which mouse is female and which is male. So I don't necessarily know if there's gross anatomical differences between the two, but there are, as I mentioned previously, there are some BDNF changes in that parallel the estrous cycle, and there's also known to be like BDNF dependent forms of LTP from OSSI cells. So there could be some effects of that, but I'm just speculating at this point. But there could just be innate differences between males and females that could contribute to these types of behaviors. But there's also times where we see the anxiety was more prominent in males than females. So more work needs to be done, but I think it's important to consider both sexes.
Justin, if I may, so you talk about GenRSI. A paper that came out in GenRSI, I think 2 weeks ago, really looked at that and the effect of the stresses that you use for learning and memory in terms of footcheck and whatever, and how they influence males and females differently in terms of global hippocampus. We didn't look specifically at MOSI cells in an estrogen-dependent manner, and in alignment with what Helen published eons ago, and not in alignment with what most of the world thinks high levels of estrogen, which are present in males and in particular phases of the estrous cycle, actually disrupt the ability to learn in this type of context. So you might want to look at that, because I've seen, sure. I noticed that your female populations were almost segregated into two subcomponents there, almost the two populations.
We did not control for cycling just because it's so time consuming, but exactly. That's a fantastic suggestion. Thank you very much, Tally.
Sorry, okay.
Cool. Are there any more questions? Going once, twice? Okay. Well, if you have a question, contact Justin. He put his email up. The talk is on YouTube, and you can get the link and watch it again if you want. Thanks, Justin. You did a great job. I think you should be proud of the work you did, in New York, and best of luck in Toronto.
Thank you very much.
For your future work, and thanks for giving us a talk.
Great to see everybody. Hope to see you soon.